Hey guys, congratulations on getting through the midterm last week. We are now moving forward. This is week 7, and we are going to be talking about the lymphatic or lymphoid system and our immune systems. So the lymphoid system is a system including cells, tissues, and organs that are responsible for defending the body against internal and external threats. We have mechanisms for both non-specific and specific defenses, and we're going to look at each of those and how separate they are. The immune response provides a specific defense, so that's the last thing we're going to look at today. The term immunity means the ability to resist infection and disease through the activation of these specific defenses. The lymphatic system has multiple functions. Draining excess interstitial fluid from tissues. We learned about fluid coming out of the capillaries into the tissues, creating interstitial fluid and interchanging solutes and ions between the blood and the interstitial fluid, but that interstitial fluid needs to be drained because we also learned that there's more fluid that's lost from the capillaries than that comes back into the capillaries. So that additional fluid needs to be drained out of the tissues and it drains into the lymphatic system. It also transports dietary lipids. We're going to learn about special lymphatic vessels within the digestive system for transporting lipids, and it helps to facilitate the immune response. Fluid always flows in one specific direction. We learned about the flow within the blood vessels 2 weeks ago, but the fluid that comes out of the capillaries so there's blood within the capillaries, and then there's fluid that's lost from the capillaries, which becomes interstitial fluid while it is within the tissues. That interstitial fluid drains into lymphatic capillaries and becomes lymph. And then we've got lymphatic vessels and lymphatic ducts, which are larger as you go along, and they then drain back into the subclavian veins, where again that rejoins the circulatory system and becomes blood again. So it starts as blood, interstitial fluid, then lymph, then back into the blood system. Primary cells of the lymphatic system are lymphocytes. We learned about these when we learned about blood, but we're going to learn a lot more detail about them today. They afford the ability to resist and overcome infection and disease. They respond to abnormal cells within the body, like cancer cells, and they also respond to foreign proteins, so any kind of bacteria, viruses, anything that can get in the body that doesn't belong there can be attacked by these lymphocytes. They are physical and chemical attacks render the threats harmless, ideally. So they want to go and attack, say, a bacterial cell, and they want to kill that bacterial cell before it spreads and creates an infection in the body. Non-specific defenses are anatomical barriers and defense mechanisms that slow the entry of infectious organisms into the body. So these are innate, you are born with them. So they include the skin, which has that keratin layer, desmosomes, a basement membrane. They also include glandular secretions. So you've got sweat and secretions that are secreted out onto the skin to help drain bacteria away from the skin. And then hair, which helps to protect your eyes and other mucous membranes from invasion. So they all work to keep pathogens out of the body. It also includes a cell called the natural killer cell, which is a phagocytic cell, and it's a type of lymphocyte. We're going to learn more about that later on. Components of the lymphatic system include lymph, which we just discussed. Lymph resembles plasma in its concentration. However, it has a lower concentration of suspended proteins. Remember that there are only small proteins that can actually leave capillaries. Those larger proteins are kept within the cardiovascular system, and so the lymph or lymphatic fluid has a lower concentration of proteins in it. We have a network of lymphatic vessels, and if you look at this picture, it looks similar to what we would see in the cardiovascular system of our blood vessels. So lymphatic vessels do tend to run along with veins and arteries. And then we have lymphoid tissues and lymphoid organs. We're going to learn about each of those. So like our tonsils and our spleen and our appendix. And then the cells are lymphocytes and smaller phagocytes. The immune function of the lymphoid system is to produce, maintain, and distribute these lymphocytes. Lymphocytes are derived from hemocytoblasts in the red bone marrow, and then they go to these different lymphatic tissues where they mature and become the final cells that they're going to be. So the lymphatic tissues like the tonsils, and the lymphoid organs like the spleen, the thymus, the appendix. Lymphocytes and other immune cells circulate within the blood, and they can cross capillary walls and go into the lymphatic system. So they are constantly circulating throughout the body. Our lymphatic vessels are bringing lymph, draining out interstitial fluid, and then bringing the lymph back to our systemic circulation. So we start with lymphatic capillaries, and then those drain into small lymphatic vessels, which would be like venules, and then into major lymph collecting vessels, which would be more like veins, and then those veins drain into ducts. They're not veins, the major lymph collecting vessels drain into lymphatic ducts. Lymphatic capillaries are similar in size to our cardiovascular capillaries. However, they originate as pockets. So you can see here, maybe I'll make that bigger like that they start as this pocket. They're not an open-ended tube. They're closed on one end. And so they start like that, and they're not like blood capillaries are open. They're just tubes that connect arterioles to venules. But our lymphatic capillaries begin as these pockets and then drain into the larger vessels. They're flattened or irregular shaped, more like a vein than an artery, and they have very thin walls, so thinner walls than capillaries within our cardiovascular system. The structure of their cells is really interesting. So the cells are overlapping, and they only allow fluids to enter the lymphatic capillaries, but not to leave it again. So it's like a one-way transport. So they can collect viruses and bacteria and debris from the tissues, and then it gets trapped in that lymphatic capillary, and then it has to move through the lymphatic system where it gets presented to a lymphoid tissue or lymphoid organ, and it traditionally or typically would be then sort of recycled by the lymphocytes in that region, so in the lymphatic tissue. Lacteals, this is what I was talking about earlier, are prominent lymphatic capillaries within the small intestine, and those are important for the transport of dietary lipids. So that is something that I would like you to know. Lymphatic capillaries are absent in areas that don't have a blood supply. That makes sense, because if you don't have a blood supply, you don't have excess fluid coming out of the capillaries, so there's not excess fluid to be collected back into lymphatic capillaries. They're also absent in the bone marrow and the central nervous system. One other thing is they appear golden in color under the microscope. The small lymphatic vessels are sort of comparable to our veins. They've got 3 layers of their walls have 3 layers like veins, but the pressure within the lymphatic system is really minimal, so they do have valves, and the valves help with propelling the lymph in the direction that you want to go. The valves are pretty close together in lymphatic vessels, so they kind of make like a beads on a chain like appearance in these lymphatic vessels. So if you can see up here, this would be like one bead, two beads, three beads. So they get this kind of beaded appearance because they've got valves that are very close together. We have two sets of our major lymph collecting vessels, superficial lymphatics and deep lymphatics. The superficial are exactly where they sound like they would be. They're in the subcutaneous layer under the skin, in areolar tissues of mucous membranes and of serous membranes. And then our deep lymphatics are larger vessels that accompany our deeper arteries and veins. So they're draining skeletal muscle and organs. So like the pericardium, the digestive system, the urinary system, reproductive tracts, all of those are being drained by these larger lymphatics. And then those larger lymphatic collecting vessels are going to converge to form lymphatic trunks. The trunks empty into two of the largest collecting vessels, the thoracic duct and the right lymphatic duct. The thoracic duct, as you can see in this darker green, collects lymph from the body, from the diaphragm, everything below the diaphragm, and from the left side of the body above the diaphragm, so it's much larger. And then the right lymphatic duct collects lymph from the right side of the body superior to the diaphragm. Again, the thoracic duct is going to drain the left side of the head, neck, chest, upper limbs, and the entire body inferior to the ribs or inferior to the diaphragm. And it's going to drain into the left subclavian vein. It begins inferior to the diaphragm around L2. And where it begins, there's an expanded chamber called the cisterna chyli or chyli. So the cisterna chyli is the origination of the thoracic duct around L2. It receives lymph from that whole lower part of the body, and then it gets pumped up into the up through the thoracic duct and is dumped into that left subclavian vein. The right lymphatic duct, again, is collecting lymph from the right side of the body above the diaphragm, so the head, the chest, and the right arm, and it's going to empty into the right subclavian vein. That's all I want you to know. You do not have to memorize those different trunks there. Something I wanted to touch on in the last slide that I missed was lymphedema, which is the collection of lymph in subcutaneous tissues. That happens due to an obstruction in a lymphatic vessel, so you end up with swelling in that region. If that's not treated and you can't drain that lymph, it can exceed the elasticity of those tissues and become a constant, like long-term problem. And if there's no drainage in that area, then you're not draining out any of the bacteria or viruses or anything that might get stuck there. So you're at a much higher risk of infection in tissues with lymphedema. So lymphocytes make up 20 to 30% of our white blood cells. We learned that a couple weeks ago. And we have three different classes of lymphocytes: T cells, B cells, and natural killer cells. So your body, the average adult body, contains over 2 pounds of lymphocytes. I find that to be pretty fascinating. This is showing us where our lymphocytes come from. So we have our red bone marrow, and we start with the hemocytoblasts. Those are going to differentiate into lymphoid stem cells. So all of our lymphocytes are going to come from lymphoid stem cells. And then some of those lymphoid stem cells are going to go over to the thymus, where they're going to create T cells. So T cells are thymus-dependent lymphocytes. And then other ones are going to create B cells within the red bone marrow. And the last are going to create natural killer cells. So natural killer cells are for immunological surveillance, meaning they go out into the body and they are always looking around for infections, bacteria, viruses. They're looking for cancer cells. So they're like our immune surveillance cells. B cells are for antibody-mediated immunity. So B cells are going to create all of the antibodies that we have to fight off multitudes of different infections. And then T cells are for cellular-mediated immunity, where cells attack other cells. We have three different types of T cells. Our cytotoxic T cells, which attack foreign cells or body cells that are infected with viruses. And those attacks are typically direct contact. So that's a cell-mediated response is with our cytotoxic T cells. And we often, if you see a capital T with a little c like that, that indicates, that looks a bit like an L, but that would indicate a cytotoxic T cell. Oops. T helper cells are used to stimulate both T cells and B cells. So T helper cells are very important in stimulating B cells. Without the stimulation of a B cell by a T helper cell, you cannot make antibodies. So that's very, very important. And then our suppressor T cells inhibit the activation of T and B cells, and so they're helping to keep your immune system in check so it doesn't kind of run away with itself. So helper cells and suppressor cells both serve that regulatory role to make sure that we're sensitive but not overly responsive in these different immune responses. The T cells are the fastest cells, so they often will get to an infection quickly. B cells make up about 10 to 15% of our lymphocytes. When they're stimulated by a T helper cell, they differentiate into a plasma cell, and the plasma cells make our antibodies. Antibodies are also called immunoglobulins. We're going to learn about each of the different types of antibodies a little bit later. Those antibodies bind to specific targets called antigens. Remember when we were learning about blood types, we learned about the A, B, and Rh or D antigens and how we have those on our individual red blood cells. And then if you have the A antigen, then you also have the anti-B antibody. So those antibodies are all created from plasma cells, and the plasma cells are derived from B cells. Once an antibody binds to its target, so it finds a cell that has its specific target antigen on it, it's going to start a chain reaction that's going to end in the destruction of that cell. And we learned about that when we learned about blood transfusions, and if you were to give somebody with type A blood a transfusion with type B blood, the type A body with the anti-B antibodies would end up killing or agglutinating all of the type B blood that was brought into their system. So the B cells are responsible for antibody-mediated immunity or humoral immunity. Humoral immunity is within the body fluids because the antibodies only occur within the body fluids. They don't leave the bloodstream. Our natural killer cells make up 5 to 10% of our lymphocytes. They're also called large granular lymphocytes, and they attack foreign cells, normal cells that are infected with viruses, and cancer cells that you find in normal tissues. They are the police, or that immunological surveillance, so they're always traveling throughout your system looking for foreign cells or infected cells that need to be removed. These are part of the non-specific or innate defense, and I always think of it as innate and natural killer. So if you look at the ends, then the natural killer cell is the cell of the innate defense. That's the lymphocyte that's involved in innate defense. The other ones are involved in the specific defenses. This is a review. We've seen this before, basically showing that the hemocytoblast is going to differentiate into lymphoid stem cells within the bone marrow. Some of those will travel to the thymus and become T cells. Other ones will stay within the bone marrow and become B cells, which will go to the spleen and the lymph nodes, or natural killer cells, which are going to circulate throughout the tissues of the body. So we're going to look at different lymphoid tissues. So these are connective tissues that are dominated by lymphocytes, and then we find these throughout the body. A lymphoid nodule is a cluster of these densely packed lymphocytes that's within an area of areolar connective tissue. So lymphoid nodules do not have a capsule. Examples of lymphoid nodules are the tonsils, linings of the digestive and urinary system, the reproductive system, the respiratory and urinary systems, and also within lymph nodes and in the spleen, we've got lymphoid nodules. The center of the lymphoid nodule is called the germinal center, and that's where you've got this lots of dividing lymphocytes in the center of that lymphoid nodule. MALT is called mucosa-associated lymphoid tissue, and this is a collection of lymphoid nodules linked with the digestive system. So an example here would be what's called a Peyer's patch. So Peyer's patches are clusters of lymphoid nodules that are deep to the epithelial lining of the intestine. And so they're like an aggregation or a cluster of nodules, and you can see that here. So that's within the digestive system where it's Peyer's patches. And then the appendix, which is also called the vermiform appendix, has a mass of fused lymphoid nodules within the wall of the appendix, and that is at the junction between the small and large intestine. So the appendix has some capabilities of helping with immunity in the digestive system. Our tonsils are large lymphoid nodules in the walls of the pharynx. Most people, when we think of tonsils, we just think of the two palatine tonsils that you can see when you open your mouth. So we have a left and right palatine tonsil. The adenoid is a pharyngeal tonsil. You can see that that's up higher, up here. The palatine tonsils are here. And then there are two lingual tonsils, which are underneath the tongue, and those are only visible if they're swollen, but typically they're small. When you get a swollen lymph node, it means that lymph node is working to clear an infection from the body. The tonsils are largest at puberty, and then they begin to sort of atrophy and decrease in size as you get older. Lymphoid organs are surrounded by a fibrous connective tissue capsule. So what makes a lymphoid nodule different from a lymphoid organ is that capsule that surrounds the lymphoid organs. So our lymph nodes, our thymus, and our spleen are our main lymphoid organs. Lymph nodes are the smallest lymphoid organs. They range between 1 and 25 millimeters in diameter, which is very small. They're surrounded by a fibrous capsule, which is what makes them a lymphoid organ. And they have trabeculae, which you can see here are partitions between the different internal sections of the lymph node. The hilum is this area here where you've got the blood vessels entering and leaving the lymph node. And then you've got lymphatic vessels. So the lymphatic afferent, you can see down at the bottom here, is bringing lymph to the lymph node. And then that lymph is going to filter all the way through from the cortex through the paracortex and the medulla, and then as it gets filtered through, all of the pathogens that are inside of it are being broken down or attacked by these lymphocytes, and then they're going to leave via the efferent vessel that's at the hilum. So lymph enters the lymph node opposite from the hilum, filters all the way through the lymph node where it's getting cleansed of any foreign pathogens, and then it leaves through the efferent vessel at the hilum. This helps to purify the lymph before it gets entered back into the general circulation. The thymus is an organ located in the mediastinum. You can see that it's superior to the heart, between the lungs, and it's posterior to the sternum. It's got two lobes, so it has a left lobe and a right lobe. And it has septa, which are fibrous partitions that divide those lobes into little lobules. So you can see how it looks like it's globular. So those lobules have a cortex and a medulla, which is called Hassel's corpuscles. The function of the thymus is to produce T cells. It's growing and functional in children. It reaches its maximum size at puberty, and then after puberty, it starts to decrease in size, which is called involution. And you can see that here, involution. As it decreases in size, it also loses its secretory ability, which decreases the function of your T cell mediated immune response. Thymosin is a hormone that's produced by the thymus that helps to promote the development and maturation of lymphocytes. So as it involutes, it is going to be producing less thymosin, so you're going to have decreased development of new T cells. The spleen is the largest collection of lymphoid tissue in the adult. You can see it here in purple. That's our spleen. It's on the left side of the body. And the spleen's function is to filter the blood. So it removes abnormal blood cells and other blood components through phagocytosis. It also stores iron that gets recycled from red blood cells. We learned about that when we discussed blood. And it helps to initiate the immune responses by B cells and T cells. When there are antigens within the blood, they can get detected within the spleen and then stimulate T and B cells to start creating plasma cells and antibodies. So when there are circulating antigens, they can get picked up in the spleen, stimulate an immune response. The spleen is surrounded by a capsule. The pulp are the cellular components of the spleen. You've got red pulp and white pulp. The red pulp has a lot of red blood cells and macrophages. The white pulp is more of a lymphoid tissue. So the blood is going to pass through the white pulp and then through the red pulp and then into sinusoids, which are going to empty into small veins that will leave the spleen. The spleen is really fragile, and so it can be ruptured easily. When it gets ruptured, it's very difficult to repair because it's so fragile. So oftentimes that's going to lead to a splenectomy where the spleen gets removed. You can live without your spleen, but you're going to end up having a decreased immune function when the spleen is not working. So we're going to go over all of our different non-specific defenses before we move into our specific defenses. So we already talked about this first line of defense being physical barriers like the skin, hair, sweat glands, secretions, all of those things. Number two is our phagocytes. Phagocytes are free and fixed macrophages, neutrophils, eosinophils, and monocytes. Remember, neutrophils are the most numerous and they are the fastest to respond to an infection. And all of the phagocytes together are termed the monocyte macrophage system or the reticuloendothelial system. So that's a diffuse collection of phagocytes throughout the body. So the monocyte macrophage system or the reticuloendothelial system. Those are interchangeable terms for the collection of phagocytes that we have in our body. Immune surveillance by the natural killer cells is number 3 here, and we're going to look at that in more detail in just a minute. Interferons can be released from specific cells activated by They can be released by activated lymphocytes, macrophages, or virus-infected cells, and they increase the resistance of specific cells to viral infection. So in the presence of interferon, cells are less likely to be infected by a virus. The complement system we're also going to look at more closely. That helps to break down cell walls and rupture these or lyse these pathogens. The inflammatory response has multiple effects. This should be a review from AMP1. That the mast cell is going to release histamine and heparin. It's going to increase blood flow, activate phagocytes, increase capillary permeability, activate the complement system. Blood clotting system is going to come in and wall off the region so that infections can't spread. Temperature is going to increase, and specific defenses can also be activated through the mast cell, and we'll see how that happens a little bit later on. And last, we have fever. Fever helps to mobilize defenses and accelerate repairs, and it also inhibits some pathogens. When you have a fever, there are certain pathogens that live only at body temperature. So if you increase your body temperature above the level where those pathogens can comfortably live, it can end up killing those pathogens. So here's our natural killer cell activation. The first step is recognition and adhesion. So the natural killer cell is going to find this abnormal cell and adhere to it. Then the Golgi apparatus is going to realign so that it is pointing toward that irregular abnormal cell. Then the Golgi is going to secrete molecules of perforin or perforins. The whole purpose of perforins is shown down here. They're going to create a hole or perforate the cell membrane of the abnormal cell. So when you have holes in the cell membrane, that's going to end up leading to lysis of the cell because the cell can't keep its internal cellular components inside. They start to leak out, and the cell is going to break down and rupture. So natural killer cells secrete perforins, which are going to perforate the cell membrane of the abnormal cell and lead to cell rupture. The complement system is a system that complements or enhances the action of antibodies and phagocytes within our bodies. Complement proteins interact with each other in cascades, so it's similar to the blood clotting system where you've got these cascades that happen. Complement is the same way. So once it's activated, there's this cascading effect that's going to lead to destruction of cells. So there are three different pathways: the classical pathway, the lectin pathway, and the alternative pathway. The classical pathway is the most rapid and most effective, and it begins with complement proteins that are all numbered like C1, C2, etc. The lectin binding pathway binds the protein mannose binding lectin, or MBL, to carbohydrates on specific pathogens. And the alternative pathway utilizes proteins that are called factors. So all of these pathways function by killing the pathogen through cell lysis. So it's going to create rupture of the pathogen cell. They also can create opsonization, which is when you have these complement proteins that are covering, like you can see in this picture here, they're covering the pathogen, then that can cause it enhances phagocytosis by, instead of having a smooth outer layer to the pathogen, it can make it rough and allow the phagocytes to attach to it more easily. And they enhance inflammation through the release of histamine. So in this picture, it's showing the classical pathway. First, the antibody is going to attach to the bacterial cell wall, so that's happening here. And then we've got the C1, or complement protein, that has to attach to two separate antibodies. And that's sort of like a fail-safe mechanism, if you only attach to one antibody, nothing's going to happen. has to attach to two antibodies that are on the outside of this bacterial cell wall. Once that happens, that initiates this cascade where you've got all of these other complement proteins that are coming in and activating the inflammatory response. So that's the main purpose of complement, is to activate the inflammatory response. Here you can see that C3b is going to be binding to the bacterial cell wall and causing that opsonization where you are enhancing phagocytosis. The alternate pathway is similar. You've got C3 that starts, and it requires these factors to split the C3 into C3a, which helps to activate the inflammatory response, and C3b, which is going to adhere to that cell wall and allow for opsonization. So read a little bit closer about those in your book, just so you have an understanding of what they are. But the main thing I want you to know about complement and the complement pathways is they function to kill pathogens through cell lysis. They function in opsonization to enhance phagocytosis, and they enhance inflammation through the release of histamine. So they're really increasing the inflammatory response. So this is showing you this C3b is attaching to create opsonization so that you've got more of the ability of the phagocytes to engulf the pathogen. And then in this one here, they're forming pores. So it's similar to the perforins that we saw earlier, but these pores are called membrane attack complexes, MAC. You can see that over here. And they form pores within the cell membranes of the pathogens, and then that's going to lead to cell lysis. So the complement pathway, C3b, is for opsonization. Membrane attack complexes are for creating those pores and leading to cell lysis. Inflammation is a localized tissue response when there's injury or infection. Everybody should have noticed that if at one point you've ever had an injury or an infection in your body, it gets red, swollen, hot, and painful. So those are the cardinal signs of inflammation down here: swelling, redness, heat, and pain. And this can be stimulated by any kind of impact, abrasion, chemical irritation, infection. All of those different things can stimulate the local inflammation. Damaged cells in the area are going to release prostaglandins, proteins, and potassium ions, and that's going to help with that inflammatory response. And it can also be introduced to foreign proteins or pathogens. So cells in the area, when they notice so you have like those dendritic cells in your skin that are always surveilling. So if they notice a pathogen in the area, they can present that foreign antigen and initiate a response by the body. So here's the inflammation and the steps in tissue repair. So up at the top, we've got tissue damage up here, and that's going to lead to chemical changes in the interstitial fluid, which is going to activate mast cells. Mast cells are going to release histamine and heparin. In the blood vessels, they're going to dilate blood vessels, increase blood flow, and increase the permeability of those vessels, which is going to bring more blood to the area, more phagocytes to the area. The histamine and heparin are also going to attract phagocytes. They can also release cytokines, which can activate some specific defenses, and the phagocytes are going to remove debris. Eventually, with the blood, they're going to have clotting, which is going to temporarily wall off an infection to keep it from spreading. And in the very end stage, you're going to have scar tissue repair of the injured area. So that should be a bit of a review from A&P1 last semester. This should also be a bit of a review. So after tissue injury, things kind of get worse before they get better. There's necrosis or dying of the localized tissue. This can lead to pus and an abscess if it gets walled off in an enclosed space. That an abscess is the accumulation of pus within an enclosed space. Lysosomal enzymes are going to break down injured cells and then attack surrounding tissues, causing that necrosis, and local inflammation is going to occur until we have that scar tissue formation. Fever is also a non-specific defense. Fever is a body temperature over 99. It's regulated by the hypothalamus. Pyrogens are proteins that can reset the thermostat of the hypothalamus to increase a body temperature. And pyrogens are produced by bacteria, molds, viruses, yeasts, multiple different pathogens that can enter the body. They produce these proteins that actually tell our body to increase our temperature, and that increased temperature can trigger the death of those pathogens, which is good for us. Interleukin-1 is also released by macrophages, and that is a pyrogen as well. So fever can inhibit those viruses and bacteria, and it also increases the rate of metabolism within the body, which can speed enzymatic reaction and accelerate the tissue defenses and accelerate repair. So everything we just talked about with the inflammation, fever, complement, natural killer cells, all of those things are non-specific defenses. Now we're going to talk about specific or adaptive immunity. So specific immunity is coordinated by T cells and B cells in response to specific antigens. So all of the non-specific things are going to react to any pathogen that enters the body. So that non-specific or innate immunity is reacting to any pathogen, any and every pathogen that enters the body. Specific immunity is reacting to a specific antigen, and you need to have receptors on the cells for that specific antigen. So we have two subsets of specific immunity or adaptive immunity. So one is the cell-mediated immunity, which is coordinated by T cells. This is going to defend against abnormal cells and pathogens that are inside of cells. And then antibody-mediated immunity or humoral immunity, which is coordinated by B cells, and that defends against antigens and pathogens within body fluid. B cells cannot cross the plasma membrane, so they have to attack from the outside of the cell. This is a good figure for you to spend some time studying. We're looking at the immune system, and we have got innate immunity and acquired immunity. Innate immunity you are born with. So there's no prior exposure or antibody production involved. So everything we talked about with non-specific immunity is considered innate immunity. Acquired immunity is what we're going to be talking about coming up, which is requires prior exposure to an antigen and the formation of antibodies. So acquired immunity can be acquired in a couple of different ways. You can have active immunity or passive immunity. Active immunity is produced by antibodies that develop in response to an antigen. So an antigen will come into the body, your immune system will respond to that antigen, and then you will have immunity to that antigen going forward. Active immunity can be acquired in two different ways. It can be induced active immunity, which is artificially acquired active immunity, and that is through the administration of like vaccines. And then naturally acquired active immunity is developed after exposure. So if you have the chickenpox vaccine, you have an artificially acquired active immunity, whereas if you have the chickenpox as a child, then you have naturally acquired active immunity. Passive immunity is produced by the transfer of antibodies from one person to another. So that can also be natural or induced. So naturally acquired passive immunity occurs with the transfer of antibodies from the mother to the baby across the placenta and then later through breast milk. Induced passive immunity is when you actually take antibodies from one person and inject them into another person. So the rabies vaccine is actually rabies antibodies that are being injected into an infected person. So take some time. I would definitely expect to see a couple questions about active and passive immunity and whether they are naturally acquired or artificially acquired or induced. So when we're talking about specific defenses, we have cell-mediated immunity, which uses T cells that are activated and phagocytes, and those are going to directly attack antigens. Or we have antibody-mediated immunity using B cells, which are going to create antibodies, and the antibodies are going to go and attack. So in cell-mediated immunity, the actual cells are going to attack other cells that have these antigens. And in antibody-mediated immunity, the antibodies are going to attach to the cells that have the antigens and aid in destroying them. Our specific defenses are specific. That's one of the properties of this type of immunity, is specificity. So you have the production of antibodies in response to the molecular structure of an antigen. So either a T cell or a B cell is going to respond to a specific antigen. So like the anti-B antibody is going to respond to the B antigen on red blood cells. That happens with thousands of different organisms. So we have lymphocytes that have the specific antibodies or the ability to produce antibodies to thousands and thousands of different organisms. It's also versatile, so there's a bunch of different lymphocyte types present within the body so that you have the ability to react to multiple different situations that can occur. We also produce memory cells, and memory cells don't have any function in the initial infection or the initial response to an antigen, but they mount a huge response upon second, third, fourth, any subsequent exposure to the same antigen. And the immune system has tolerance. So with our immune system functions to attack foreign cells, but also to tolerate or ignore normal tissues. When T cells and B cells are being created, if they begin to attack any of our normal cells, they're going to be destroyed. And up to 98% of developing T and B cells are destroyed because they react to normal tissues. So this tolerance is where we want our immune system to let our normal body be itself, but recognize foreign antigens and attack those. In order for an antigen to be detected by the body, it needs to be presented on the cell membrane of a cell. And it is presented in what's called a major histocompatibility complex antigen. So they're also called human leukocyte associated antigens, or HLA. So these are proteins that are on the cell membrane which can take an antigen and present it to other cells within the body. The MHC antigens are unique to each individual, except in identical twins, they would have identical MHC antigens. The purpose of these markers is to differentiate self, or what's sort of normal, from non-self. So having an empty MHC protein is going to look normal, but it can also pick up pieces of a pathogen that have been engulfed by that cell and take that pathogen and present it on its cell membrane. There are two different classes of these MHC antigens. Class 1 is built into the plasma membranes of all cells within the body except for red blood cells. And class 2 are on the surface of antigen presenting cells only. So also our T cells that have been activated by exposure to an antigen, so our antigen presenting cells are all of our phagocytic cells and our dendritic cells. So those all have class 2 MHC proteins, whereas all other cells in the body have class 1 MHC proteins. So this is showing you the MHC1, and this is the actual MHC protein on the cell membrane surface. So what happens is we start over here with our viral or bacterial infection, comes into the cell, it releases these abnormal peptides, and those peptides are picked up by the MHC1 antigen, and it goes through the Golgi apparatus into a transport vesicle where it gets deposited onto the cell membrane, and it's going to display that abnormal peptide in that MHC1 protein antigen. Type 2 is going to be very similar. This cell is a phagocytic cell, so it's going to engulf the entire protein, break it down with a lysosome, and then the MHC2 protein is going to pick up the antigens from the lysosomal breakdown of that cell, and then it's going to display the fragment of that antigen in the MHC2 protein on the plasma membrane of that antigen presenting cell. In addition to those MHC proteins, there are also these cluster of differentiation proteins, which are membrane proteins that are found in lymphocytes, macrophages, and other immune-related cells. So antigen recognition includes the binding of a T cell to its specific target, and that binding happens at these cluster of differentiation proteins. There are over 350 different cluster of differentiation markers, but the two important ones for talking about T cells binding are the CD8 marker, the CD8 cell, and the CD4 marker. So the CD8 marker is found on cytotoxic T cells and suppressor T cells, and they respond to antigens in the class 1 MHC protein. The CD4 marker is found on T helper cells, and they respond to antigens in the class 2 MHC protein. The way that I remember this is if you multiply the MHC protein class by the cluster of differentiation marker, it always equals 8. So the CD8 times the class 1 protein equals 8, and the CD4 times the class 2 protein also equals 8. So CD4 connects to class 2 proteins, CD8 connects to class 1 proteins. So let's look at this a little bit more closely. So here we've got an infected cell, and it is presenting its antigen on this class 1 MHC protein. So you can see that that MHC protein is on the infected cell, and it is presenting this antigen. So we have this cytotoxic T cell that comes along, and it has a receptor for that specific antigen. So in order for it to attach to this antigen, it also must have this CD8 marker. So it's the CD8 marker is allowing the T cell to attach to the class 1 MHC protein that has the antigen that is specific to the receptor on this T cell. If the MHC protein or the antigen that's being presented is not the specific antigen that that T cell is sensitive to, the T cell will move on. So this is where that specific immunity comes in, is only the T cell with the specific receptor to the specific antigen that the infected cell is presenting is going to go and connect there. Once the T cell actually connects in here and binds, then that's going to activate that T cell. The activated T cell is going to start dividing. It's going to create these active T cells, which are going to be cytotoxic T cells, which can then go and bind and destroy those infected cells. And it's going to create memory T cells. Memory T cells are going to remember this so that next time they see that antigen, they can activate immediately and mount a huge response. Now here we've got an infected cell, and we have this inactive T helper cell. So the T helper cell has the CD4 protein, and it's going to go and bind to this class 2 MHC protein on the infected cell. So the first one we just looked at on the last slide was a class 1 cell with a cytotoxic T cell. Now we have a class 2 MHC protein with a T helper cell, and the recognition protein is that CD4 protein. So that's what allows this binding to occur. Once that binding happens, we've activated the T helper cell, and we've created memory T helper cells. The activated T helper cells are going to go and secrete cytokines, which are going to stimulate B cells and create this antibody-mediated immunity that we're going to talk about soon. The memory cells are inactive until a subsequent exposure to the same antigen takes place, and then the memory cells activate and mount a giant response. So just to recap here, we've got the infected cell, which is going to have either the class 1 or class 2 MHC protein. And you can see that there's a little bit of a difference in the shape between the MHC1 protein and the MHC2 protein. When it is infected, that protein is going to present a portion of that antigen on its cell membrane. Once it's presented, it's going to bind to and then activate this CD8 T cell. This is the MHC1 protein is going to activate this CD8 cytotoxic T cell. So once those cytotoxic T cells are activated, they're going to attack and destroy infected cells. It's going to create memory T cells, which are going to await that reappearance or subsequent appearance of the antigen. And it's going to also stimulate suppressor T cells to control that immune response. Over on the right here, we've got the class 2 MHC protein. It's presenting its antigen to the CD4 cells, which is the T helper cells. So it's going to stimulate T helper cells, which will in turn stimulate B cells to produce antibodies. And it's going to create memory T cells, which will await that subsequent appearance of the antigen to mount a bigger response. So remembering that the MHC1 protein is going to connect with the CD8 on the cytotoxic T cells, and the MHC2 protein is going to connect with the CD4 protein on the helper T cells. Now we're going to move on to B cells and antibodies. So when a corresponding antigen to a specific B cell antibody appears in the interstitial fluid, it's going to react with the antibody that's on the membrane of the B cell. That's going to sensitize the B cell by binding that antigen-antibody complex, and this typically happens in a lymph node near the infection site or the injury site where this pathogen has entered into the body. B cell membranes have class II MHC proteins, and the antigen is taken into the B cell by endocytosis. And so then that B cell is put on standby, so it's ready to be activated, and a T helper cell has to activate the B cell by binding to the MHC complex. Once the B cell is activated, it's going to start dividing and making plasma cells. So it's going to divide and make active cells and memory B cells. So let's look at this in pictures. I always think pictures are easier than words. So step one over here is sensitization. So the B cell is getting sensitized. So we've got this B cell, and these antigens are going to come and bind to the antibodies on the B cell. So antibodies are Y-shaped like that. So you can see there are two antibodies, 1, 2 here, that are on the outside of that B cell, and so they are both binding to these antigens. The antigens are taken into the B cell, and then they're presented in the MHC protein on the membrane of the B cell. So now the B cell is sensitized and it's presenting its antigen. The sensitized B cell is going to connect or bind to a T cell. When that happens, the B cell gets stimulated, cytokines are released, and the B cell is going to do a couple of things. One thing is it's going to make memory B cells, which are clones of itself. So those clones are all going to be sensitive to that same antigen. And the other thing it's going to do is make activated B cells. Activated B cells differentiate into plasma cells, and plasma cells are going to create all of these different antibodies. So an antibody is Y-shaped, so it's got the sort of the post and then the two parts coming up, so it's shaped like a Y. And it's got two heavy chains and two light chains. So the heavy chains are these longer chains like that, and the light chains are these shorter chains. Where the heavy chains and light chains come or end is the antigen binding site. So antibodies are going to bind to antigens at the antigen binding site. These antibodies also have complement binding sites. So when they get activated, they can activate the complement system as well. So let's look at all the different classes of antibodies. So remember, they're called immunoglobulins. So we have IgG, IgE, IgD, IgM, and IgA. IgG is the largest and most diverse class of antibodies. It makes up about 80% of the antibodies, and the IgG is the only type of antibody to cross the placenta. An example of an IgG antibody is the anti-Rh antibody. So remember when we were looking at blood types, Rh, you're either Rh positive or Rh negative. IgG, since it crosses the placenta, the anti-Rh antibody can cross the placenta and attack an Rh positive newborn during the second pregnancy with an Rh negative mother. Remember, that's the hemolytic disease of the newborn. IgE antibodies are found, they are attached to a molecule that's exposed on basophils and mast cells. And so we find them with allergies, and they increase inflammation. So IgG are related to allergies. So the allergic response creates IgE antibodies. IgD antibodies are on the surface of B cells where they can bind antigens in the extracellular fluid, and this can plays a role in the activation of B cells. So IgD helps to sensitize or activate B cells. IgM has this interesting like star-shaped structure, and IgM is the first class of antibody or immunoglobulin that gets secreted after an antigen arrives. So once you have an antigen in the system, IgM is going to get secreted first and quickly, or it's going to get created more quickly, and then IgG is going to come along later. So we're going to look at that in a minute. But the IgM, so examples of IgM immunoglobulins are the anti-A and anti-B immunoglobulins when we're looking at blood type compatibility. So those are IgM immunoglobulins. And then last are IgA immunoglobulins, and those are found mostly in glandular secretions like mucus, tears, saliva, and semen, and breast milk as well. So the IgA is secreted into breast milk, and that offers some protection to the newborn while breastfeeding. There are 7 different ways that an antigen-antibody complex can eliminate an antigen. The first is through neutralization, where the antibodies bind to the target regions of the antigen, like the virus or the bacteria, and prevent them from attaching to cells. So then they would just get circulated out of the system because they're neutralized and they can't attach to your cells and cause infection. The second way is through precipitation and agglutination. So basically, the antibodies can grab onto sites on multiple different antigens, creating a molecule that's too large to remain in solution. So you get this big insoluble molecule that precipitates out. Remember when we were looking at the blood typing, there was agglutination with anti-A antibodies. When you put them in with type A blood, the red blood cells would all clump together. So that's what agglutination is. The third way is through activation of complement. Remember, we had those complement binding sites on the antibodies. So when they bind to an antigen, they can change shape, expose the complement binding site, and then activate complement, which the complement can come in and destroy the antigen that is connected to that antibody. They also work to attract other phagocytes. So antigens that are covered in antibodies attract eosinophils, neutrophils, and macrophages to come and engulf those antigens. They work through opsonization. So when you coat a pathogen with antibodies, it makes phagocytosis of that pathogen more effective. Stimulates the inflammatory response through stimulating basophils and mast cells. And last, prevents bacterial and viral adhesion. So IgA antibodies that are dissolved in saliva, mucus, other secretions can make it difficult for pathogens to attach to the body and penetrate body surfaces. Our body responds differently the first time it comes into contact with an antigen than it does the second time. So the primary response, the antigen has to activate the B cell. The B cell must differentiate into a plasma cell. The plasma cell has to differentiate and start secreting antibodies, and we're going to have a sustained but gradual rise in our antibody levels. An antibody titer is basically looking at the number of antibodies that are within the blood. And that titer with the primary response is going to peak 1 to 2 weeks after the initial exposure. So by that time, you've gotten sick and had this response to whatever this antigen is. Remember, IgM antibodies are going to be produced first, but they have a less effective response than the IgG antibodies. The IgG are going to be slower to respond, but more effective in the long run. So after 1 to 2 weeks, your antibodies are going to start to really fight off this infection and you're going to get better. The secondary response is very different. So during that primary response, you've created these memory B cells, and those memory B cells are sensitive to the same antigen that the primary B cells were in the initial response. So when you have a subsequent exposure, or the antigen comes into your body for the second time, the response through those memory cells is immediate, and activation can occur at low antigen concentrations. So you don't have to wait for the differentiation of the plasma cells and all of that to happen slowly. It happens really, really quickly. So B cells get activated, they immediately differentiate into plasma cells, and those plasma cells start secreting a massive quantity of antibodies. Your titers are going to rise very rapidly with that second infection and reach much higher levels than they ever reached during the first infection. Memory cells can survive over 20 years, so you're going to have this hardy response to a subsequent exposure for many, many, many years to come after your initial exposure. That rapid secondary response is the reason why immunizations work, because with the immunization, you have the initial reaction. So you might have a mild case of the sickness that you're being immunized against, or it might just create your body to create these memory B cells. And then when you're exposed later, so say to chickenpox, if you have the chickenpox vaccine, you're going to start creating memory B cells to the chickenpox antigen. So when you're exposed to chickenpox later in life, your body is going to rapidly fight it off and keep you from getting sick later on. So here we're looking at the primary versus the secondary response. So in the primary response, you can see that IgM is secreted first, and it has sort of a smaller effect. And then IgG is created a little bit later, but it has a greater effect than the IgM. So after about that 2-week mark here, you're starting to really fight off that infection and get better. The secondary response, you have a more rapid IgM response, but it's still small, but you have this incredible IgG response that is much, much greater than any response that you had during that first exposure. This chart is showing you the overall reaction of the body during a bacterial infection. So first thing to fight off bacteria are the neutrophils. Neutrophils are the most numerous white blood cell, and they are the fastest to respond. Second, we have natural killer cells because they're always surveilling throughout the body, and so they're going to react to a bacteria. Third, we're going to have macrophages. So the macrophages are the green line down here. Those are going to come in and start cleaning up the area. And the cytotoxic T cells are going to come in as well. So that's our cytotoxic T cell line. While all that is happening over the first week, we have plasma cells that are slowly mounting a response, and they start to secrete antibodies. So our antibody titer is going to begin week 1 and kind of max out between weeks 2 and 3. So while this B cell reaction is occurring slowly in the background, we still have our neutrophils, natural killer cells, macrophages, and cytotoxic T cells that are fighting off the the bacteria in the beginning. So this is showing you exposure to a bacteria is going to cause macrophage activation. The macrophages are antigen presenting cells. So once they present the antigens, it's going to activate cytotoxic T cells, which will go and directly attack the bacteria. They're also going to activate T helper cells. The T helper cells are going to activate B cells. The B cells are going to produce plasma cells. The plasma cells are going to produce antibodies, and the antibodies are going to go and fight those off. So you can see how the cytotoxic T cells are going to be a faster response, but the antibodies are going to be a long-term response, and they're going to create memory cells, which are going to have a much more rapid response the second time we have exposure. So this is bacterial defense. Viral defenses, the virus is going to infect our tissues, infect cells. So viruses go and they infect specific cells. Those cells are going to present the antigens on their plasma membrane. They're also going to release interferons. Interferons are going to increase resistance of other cells to those viruses so that surrounding cells, it's harder for them to get infected with the viruses. The antigen appearance on the plasma membrane of the infected cell is going to stimulate natural killer cells to destroy the cell and activate cytotoxic T cells to destroy the cells. And then they're also going to be taken up by phagocytes, our antigen presenting cells, which will present the antigens, activate the T helper cells, which is going to in turn activate B cells. The B cells are going to produce antibodies, and they're going to produce memory cells. The antibodies are going to go work on destroying the viruses that are left after all of this has occurred, and then the memory cells are going to fight off subsequent infections. This one is showing us, here's our antigen. The presence of an antigen is going to stimulate nonspecific defenses. One of those nonspecific defenses is the complement system, which is going to attack the antigen directly, and the natural killer cells and macrophages, which will directly attack the antigen as well. While that's all happening, that antigen has also stimulated our specific defenses. So remember, nonspecific is complement, natural killer cells, macrophages in response to an antigen. Specific defenses includes activation of T cells, which are going to produce memory T cells and cytotoxic T cells. Cytotoxic T cells are going to go and directly attack the antigens. And then we're also going to have activation of B cells through the T helper cells. The B cells will produce memory B cells and plasma cells. The plasma cells will produce antibodies. The antibodies will go and attack the antigens. So I would recommend that you look through all of these and make sure that you understand the difference between triggering non-specific defenses and triggering specific defenses, activating cytotoxic T cells to directly attack cells with antigens, and activation of B cells, which is going to end up making plasma cells and creating antibodies, and the antibodies are going to go and attack the antigen. Our immune system development begins very early in fetal development. About the 3rd month of fetal development is when we start to create our own immune system. IgG antibodies pass through the placenta, and then after birth, IgA antibodies pass from the mother via breast milk. So breastfeeding is, I'll put my little plug in for breastfeeding. It is very helpful and effective in helping newborns fight off disease. And then infants begin to produce their own IgG antibodies as they begin to be exposed to different infections, environmental antigens, and as they start to get vaccinated. We also have many immune disorders or disorders of the immune system. So autoimmune disorders develop when the immune system responds inappropriately to normal body cells. One ankylosing spondylitis has an HLA protein. So remember, that's the same as a major histocompatibility complex. So an HLA B27 antigen, and the body starts in ankylosing spondylitis, the body is attacking specifically that HLA B27 antigen, and it creates breakdown of the spine, and the spine begins to fuse together. Other autoimmune diseases like rheumatoid arthritis, psoriatic arthritis, lupus, that's all the immune system attacking otherwise healthy connective tissues within our body. Immunodeficiency diseases occur when the immune system fails to develop normally or when the immune response gets blocked in some way. So AIDS, the disease AIDS, destroys T helper cells, and without T helper cells, you can't activate B cells, and without B cells, you can't have plasma cells, and without plasma cells, you can't produce antibodies. So when you have somebody who's developed AIDS, they end up with a very, very minimal immune system because of the loss of T helper cells. Allergies are an inappropriate or excessive immune response to certain antigens. So I think we're going to talk about those in the next slide. Yeah, so allergens are antigens that trigger allergic reactions. So we have 4 different classes of allergies. Type 1 is immediate hypersensitivity reaction. Type 2 are cytotoxic reactions, like our ABO incompatibility. Type 3 are immune complex disorders, and type 4 are delayed hypersensitivity reactions. So we're going to talk about type 1, that immediate hypersensitivity. It's a rapid and severe response to the presentation or the presence of an antigen. So hay fever or allergic rhinitis is an immediate hypersensitivity. So if you are sensitive to, say, goldenrod, when the goldenrod comes out, your body is going to have a severe response to the presence of that antigen in your system. Sensitization occurs during the initial exposure, and what happens is you have a large quantity of IgE production. But in that initial exposure, it takes a while for you to create the IgE, so you don't have any symptoms. But then with subsequent exposure, you have sudden massive inflammation of the affected tissues. Anaphylaxis is when you have an extremely severe reaction to an allergen, and it stimulates mast cells throughout your entire body. And anaphylaxis can develop within a matter of minutes. So some of the things that happen are capillary permeability increases, so you can have severe swelling, edema, and hives can form on the skin. Smooth muscles can contract within the respiratory system, making it hard to breathe. Extensive peripheral vasodilation occurs, so that can, when you have all of that vasodilation, that can lead to a drop in blood pressure, which can lead to anaphylactic shock. You can end up going into a coma because your blood pressure drops so low. And so anaphylaxis is treated with antihistamines, corticosteroids, and epinephrine injections with an EpiPen. So here's the mechanism of anaphylaxis. So this top portion here is the first exposure. So you have the allergen that comes into the body. It's taken up by the macrophage that triggers the T helper cell, which sensitizes the B cells to create plasma cells, and the plasma cells are going to create these IgE antibodies. With the second exposure, the allergen is going to attach to the mast cell. It's going to, or attach, sorry, to this, you're going to have mast cell activation, which is going to release histamine, leukotrienes, and other mediators because those IgE, I am trying to change the color here. It's not working. Sorry, it's all in yellow. The IgG antibodies are going to come in and attack right away, sensitizing mast cells and basophils. We're going to release all of this histamine, leukotrienes, and that's going to cause capillary dilation, increased capillary permeability, airway constriction, increased mucus secretions, pain, and itching throughout your body. Long-term stress can lead to the release of glucocorticoids, like when you're in that general adaptation syndrome that we talked about. I'm not exactly sure what, an endocrine system, I think. And so that can inhibit the immune response because it decreases inflammatory response, it decreases the abundance of phagocytes in peripheral tissues, and it inhibits the secretion of interleukins, which is going to depress the response of lymphocytes. So chronic stress decreases your immune system. It also is through mast cell inhibition is what's going to decrease the inflammatory response. So chronic stress is no good for you. As we age, our immune system takes a hit, as well as most of our other systems. So the immune system becomes less effective at combating disease as you get older. T cells become less responsive to antigens. B cells are less responsive, most likely because there's decreased T helper cell activity. Without T helper cells, B cells cannot get stimulated, and then they cannot create additional antibodies. So the net result of that is an increased susceptibility to viral and bacterial infections, as well as decreased immune surveillance through the natural killer cells. So you have increased susceptibility to cancers forming and not being able to be taken care of by the immune system. That concludes our lecture on the lymphatic and immune system. If you found this chapter super intriguing, then I would recommend that you take microbiology. If you are entering your LPN year at VTC and you have any desire to move on to your RN year, I would also recommend microbiology in the coming semester. That really microbiology is required for the RN year. It is not required for the LPN year, but taking it concurrently with your LPN year, it would be a lot. So micro is super interesting, and it really builds on a lot of stuff that we learned this chapter. So I hope you guys have a great week, and I will see you back next week for the respiratory system.